Distinguished participants, I would like to welcome you. My name is Ömer Faruk. I'm from and its relation with microbiology. We will have this panel on this topic, and just in the middle of the pandemic, the world of microorganisms seems to be very integrated into our lives. It has been 2 years since the onset of the pandemic, and the previous pandemic was the Spanish flu, and after that, we schizophrenia because it was reported more after the Spanish flu, and now we're still talking about the organisms of microorganisms related to the schizophrenia, and we will be talking about the relationship between the schizophrenia and the Crohn's disease. I will make an introduction about schizophrenia and its relation with microbiology. I will be making an introduction, and the details will be given my other colleagues. I believe they will provide very important information with you. First of all, schizophrenia. After the 1980s, we know that there were more studies on schizophrenia, and with regards to the imaging and also in the field of biochemistry and with regards to neurotransmitters, there have been significant advancements, and maybe 15 or 30 years ago, we knew something different, but today we have much more different information about it. In the past, we were talking about the dopamine theory, but now we're talking about the glutamate theory. We don't know what is behind the glutamate theory, what is behind the roots of it, because we have some studies on inflammation that support this argument. One of these theories is inflammation theories, and we will be talking about today. Thank you. For the first time, there was an argument regarding the relationship between schizophrenia and the viruses, and this was argued first in 1973 by some researchers, and later on came some studies. Although there are different theories, it is not possible to talk about the consensus about it, and we believe that some information will shed light on today's perception, and influenza pandemic in 1918 is an important source of information for this. And when we look at the children who were born from the women who witnessed who actually experienced the Spanish flu in 1918 were studied, and here we see the of this theory, because it has been a long time since the influenza pandemic, and we have more advanced technologies regarding laboratory tests and PCR tests, and we also have chances to conduct studies, and it's not just about viruses but also some bacteria, parasites are investigated as well, and we have CMV, influenza, and rubella. These are the viruses that are studied by different meta-analysis, and we know that there is a relationship. I mean, we're trying to learn that there is a relationship between the microorganisms and the immune system. And of course, this is a huge puzzle, and we have only a couple of pieces of it right now, but in the future, I believe it will be clarified further. look at the recent meta-analysis, we see that there is an argument there could be such a relationship between these viruses and the schizophrenia. And when we look at these parasite studies, toxoplasm comes to the fore, and there are animal studies and human studies, blood tests or tissue tests done, and viruses and other microorganisms are studied, and we see that there is an inflammation against the viruses, against these tissues, and we know that there is an increased level of immune response and increased level of chemokines, and we know that it is correlated with the cognitive deficiencies. And in addition to these immunological elements, there is a synaptic connection, but this connection is actually disrupted by the disease. But these are all hypotheses. But the most important point here is the microglia. Dr. Ersel will be talking about it in detail. And as a hypothesis, it can be said that microglial activation actually disrupts the synaptic system, and we know that before the disease onsets, there are some negative and positive symptoms, and sometimes it's triggered by heroin or other drugs. And this is based on the microglial activity. When we look at the etiopathogenesis of the relation between schizophrenia and neuroinflammation, there are many more studies today, but what we don't know is that in the etiopathogenesis of schizophrenia, whether the immune function is the primary cause or not. And for the last 15 years, let's say, we have been seeing this, and there are lots of studies in this respect, and I mean, it's sometimes exaggerated. We have exaggerated level of studies about this, and this is microbiota. And microbial elements that are responsible for the schizophrenia may be related with the ones that are already in the flora. Therefore, in 2004, germ-free and normal rats were compared in a study, and there were some behavioral differences, and the intestinal flora and intestinal axis were analyzed. And finally, we have Dr. Ezgi, and she will be talking about COVID and schizophrenia. I tried to talk about this, I tried to mention this at the beginning, because regarding the COVID-19, we don't know the results, the consequences of them. So there could be some psychological effect of it after you recover the COVID-19, but we don't know whether there will be an increase in incidence in the future. We need time for this. But there are also some challenges faced by the schizophrenia and the mortality rate, etc. There are significant studies ongoing regarding this, and they also have difficulties with regards to their treatment. And the panelists here and I are working on this matter for a while, and I believe we will try to share the fruits we have yielded as a result of these studies. This was my presentation. It was a brief one. Now I would like to invite Dr. Ezgi Tanrıöver Aydın to deliver the second presentation. We are working at the Cerrahpaşa University Department of Psychiatry. She will be talking about COVID-19 and schizophrenia.
Hello everyone. Firstly, I would like to thank you, Professor Ömer Faruk, for the introductory speech. As he has stated, this immunological issues and clinical reflections of them are in our study field. I will try to present you in the relation in between COVID-19 and schizophrenia. First, I will be talking about the impacts on general mental health, and then what kind of impacts have been observed in schizophrenia diagnosed patients during the pandemic, or schizophrenia's importance for such pandemic times, what should be careful about, what could be the impact, and how should we manage the vaccination process. We will see the neuropsychiatric presentations as well. As an introduction, COVID-19 have been reported in China December 2019, and I've uploaded all these numbers in March 2021. There are over 5 million deaths, and we do not know the origin of the disease. However, the purpose is to control it as much as possible, curfews, stopping any gatherings, public activities, and halting business and education activities, travel bans, and also mask requirements are still continuing in different levels. The restrictions in regard to travels could be observed in the table just above. In the light of the information that we have obtained from different pandemics, World Health Organization published certain warnings. The anxiousness over disease, death, and also ambiguity cause a certain impacts in terms of psychiatry. Loneliness, anxiety, depression, sleep disorders, and tendency, idealization of suicide reported as the impact. And in the publication of World Health Organization, we should have certain lessons and get prepared for the next pandemics. And when it comes to schizophrenia patients, what kind of impacts that we have observed? As you can imagine, general population's reaction were sensed in a more intense way in this patient group. Psychotic episode increases and also tendency to suicide have been observed in the articles. There are certain symptoms comes with the disease. Unfortunately, we do not have any treatment for that. However, some compulsory interactions, saying hi to someone, shopping at the supermarket, for example, were supporting these people. However, with the social isolation, they were negatively affected, and there could be an atmosphere nurturing their symptoms in a bad way. And when it comes to the challenges of facing coronavirus disease, we should be focusing on these challenges. We had certain problems in hospitalization. It wasn't possible for them to follow the hospital. Unfortunately, they were not able to follow isolation rules, and there were restrictions. And psychiatry services are special services. We may not be allowed to open the windows. We may not place disinfectants whenever we want. And there could be, again, difficulties to follow mask requirements. Some protocols have been developed, screening tests have been developed, and with the monitoring of healthcare professionals. We have improved the situation. Regular follow-ups were also challenged because of this measures of the pandemic. And in terms of cooperation and between the doctor and the patient, it is quite important to have this interaction. But then we have used phone calls or web calls. And obviously these measures have helped these people. For the supply of the drugs, it was a problem. Then again, for the follow-up of any side effects, there were problematic issues. A suggestion, for example, if you continue with the long-acting antipsychotic diseases, any problem that the patient could experience may be diminished. I would like to highlight the issue of clozapine. Clozapine was shown as a cause of increase in infection, and there were confusing thoughts over that. Some clinicians just delayed, they postponed their plans on administering clozapine. Neutropenia in relation with clozapine was a confusing case for the clinicians. Verman et al. Had developed this new algorithm upon the questions directed to them. If you just clozapine, it could be because of viral infection and you do not have any re-administration contraindication, so you may just re-administer clozapine. Clozapine intox is another issue. In case of an inflammation, there could be increase in the blood levels for clozapine. If there is any confusion, if there is any fatigue, for example, any weirdness in the patient, then COVID-19 infected patients could get decreased level of clozapine. This will even protect the patient. But if there is a direct toxicity, you should just stop using it. Electroconvulsive treatment was another treatment that we had to postpone, unfortunately. Catatonic presentations are hugely important, and this treatment is vital for that case. Why schizophrenia is a high-risk group for COVID-19? Because with COVID-19, infection risk have increased in schizophrenia patients. Mortality rate is higher for this group, and studies have already revealed that for a person who have neuropsychiatric patient has a higher risk of catching the infection, and they have higher risk of reviews information. You may see the mortality severity studies, and also the risk of infection have been studied with these ones. And when we check the patients who have admitted to the hospital, we do see for elderly comorbid diseases could be a very common risk factor. However, for young groups, they generally have like schizophrenia. This is a very good follow-up. Nearly 20,000 patients have been included in the control group. The red one is schizophrenia patients, shows the control group. incidence the corrected risk is evaluated once again and significantly in schizophrenia patients. We do see that hospitalization and the control group, severe mental health disorders. In this group, bipolar disorder is also included, and there is a higher table. And in this one, COVID-19 tested elaborated. Schizophrenia is represented as a higher risk, and for hospitalization and mortality, higher results for being in a high risk group. Surely there should be a comorbidity, and schizophrenia patients, unfortunately, in this high risk group. Then again, for the survivors of COVID-19, when we check the results, schizophrenia patients have higher mortality rates. What kind of feature does schizophrenia when it comes to COVID-19? It could cause an impact on immune system, and it has a systemic inflammation presentation. There is a common inflammation presentation. It is continuous, and when we have COVID-19 at the same picture, unfortunately, it is quite possible to see severe presentations. And antipsychotic drugs seems to increase pneumonia risk in accordance with certain reviews. And in summary, schizophrenia patients' comorbidities, dysregulated immune response, antipsychotic use, obesity, or diabetics, or hyperglycemia increase the risk of COVID-19 mortality. How should we do the follow-up of COVID-19 infected schizophrenia patients? We should know that there is a defect and in their lives, and unfortunately there is a blood sugar regulation disorder because of their malnutrition. Polymorphism should be evaluated. AC1 disorders, for example, could be seen very quite commonly, and in COVID-19 this is a sign for severe disease. Tobacco use is another risk factor. Nearly 60 to 70% of the schizophrenia patients use tobacco. And when it comes to coagulopathy representations of COVID-19, we do see that there is a significant relation. There's an endothelial damage, and because of the stockings, there is a situation of hypercoagulable presentation, and it causes coagulation problems. Psychotic disorders also have hypercoagulation. Obesity, atherosclerosis, and cardiovascular diseases are frequently comorbidity for that group. And when these two come together, unfortunately, coagulopathy is a very commonly seen situation in that group. Neuroleptic malignant syndrome in COVID-19 patients. NMS have been reported. However, this is a schizophrenia patient, refers to hospital. It is a full capacity hospital. However, while they were following the treatment, they do observe that it is COVID-19 positive, and they interpreted it as COVID NMS. Immune modulator treatment also hugely important for the COVID-19 treatment. It causes a certain sleep disorders, delirium, mania, depression, and psychosis. And with a use of low dosage antipsychotics, these disorders and problems could be resolved in accordance with the studies. For schizophrenia patients, antipsychotic disorders have been used, and in COVID-19 services, it was a problematic case indeed. Why? Because they are prolonging QT, and they are quite open to drug interactions. This has been published by the University of Liverpool. On the left side, it is COVID-19 drugs, and on the right side, psychotic pharmaceutical agents could be added, and you can just check any interactions. And when we come to the vaccination process, this was a very good presentation. It is a one-year follow-up. For the patient group, hospitalization incidence group had 6.2 difference when we compared with the control group. However, after vaccinations, it has decreased to 1.1. This is quite good. The only problem is that their mental health should be supported by the healthcare professionals. These are the EU member and candidate countries. For example, in 8 countries, in the documents, psychiatric disorders have been mentioned in the documents for vaccination. Denmark, Netherlands, the UK have priority group for psychiatric disorders, psychosis, bipolar disorder, and schizophrenia. They had these patient groups as priority groups. Unfortunately, Turkey is not there. And COVID-19 and related neuropsychiatric presentations. When we check it, NPC is a highlight at this point. As the disease continues, as we collect the data, we notice some certain new things. It could be an acute phase in a post-infectious period, so we do not know the direct mechanism, if it is because of the virus or because of the treatment, for example. We have no idea at this point. As Omar Fark has stated, Asian influenza, and for example in Hong Kong there was another pandemic. In Spanish flu there was a severe presentation, and it was quite related to neuropsychiatric diseases, and in H1N1 narcolepsy episodes have been observed frequently. Okay, then how can we differentiate such clinical findings, subacute and chronic NPSS, in between those depression, anxiety, trauma-related disorders, psychotic disorders, Parkinsonism, and Guillain-Barré syndrome, perfect neuropathy, and myopathy could be listed for the NPSS, for post-infectious NPS, and para-infectious acute NPS. We can list loss of smell and taste, for example, could be listed in acute phase. Increased pro-inflammatory cytokines could be observed. How does it affect central nervous system through retrograde axonal transportation or heterogeneous pathway? And the disrupted cytokine system may increase pro-inflammatory cytokines intense increase, and also there could be changes in the blood-brain barrier. Microglias play an important role. How should we say? It just inflames the central nervous system, and the impact is quite long. Also, for the gut microbiota, translocation has a certain impact. ACE2 receptor has an invasion through the gut, and it causes certain changes in blood-brain barrier. This is not an up-to-date article. This is from past, but it is quite related to today. 106 patients developed new psychosis in the beginning, and half of them have affective disorder, and half of them schizophrenia disorder. When we check it, exposure to COVID-19 and IgG levels are quite high. And in another study in the UK, they have this database, neurobiologists, psychiatrists upload data to this stroke database, and we see majority of the patients have psychosis when they are referred to the hospital. Future generations schizophrenia. Here we should identify if it has a viral impact, I mean the COVID-19. There are ACE receptors in uterus. Is it the pathway to it, or is it just inflammation? Inflammation's impact on the baby. And we have the question, should we be prepared for their 20s? Will they turn into psychiatry patients? Or for the patient groups who are underage, for example, we need to be quite careful with their treatments and autoimmune diseases after the infection could be causing certain disorders like Bickerstaff's encephalitis or Guillain-Barré syndrome. These are the most commonly discussed diseases in this field when it comes to the infectious diseases. There is a very strong bond between autoimmunity and schizophrenia. And individuals who have strong autoimmunity has a higher risk of schizophrenia as well. After SARS pandemic, we have witnessed that there is a disorder in inflammatory response regulation. We have question marks in regard to the relation in between these infectious diseases and the risk of developing psychiatric disorder. Russian flu, Spanish flu, and Hong Kong flu. After these three phases, they have followed up the newborns of pregnant women who were infected in these periods in terms of psychiatric diseases and psychotic frequency. The data shows there is an increased impact. In 2019, there were H1N1 flu, and the follow-up studies are still ongoing. We do not have a data. 64, for example, there was the rubella pandemic, and it has been reported that the babies who were exposed to that had an increased rate of psychosis at the percentage of 15.7, and Zika was not reported as a disease which had a direct impact on this. And especially the types of OC43 and 229E of coronavirus had are neuropathic virus and could cause microglias infected in adults. HK HKOC43, HCOVNL63, and then again HKU1 chains are detected in the brain. So there are psychiatric disorders. There are some cases which we do see there is an impact on the brain. So we should do the follow-up very meticulously in order to observe any psychiatric disorders. Those are the things that I would like to state. Thank you. Thank you, Ezgi. It was a very nice, informative speech. And now the next speaker will be Dr. Ersat. He will be delivering the speech of how do microorganisms affect the brain.
Merhaba, Doktor Ersel ben.
Hello, my name is Ersat. I would like to thank Dr. Ömer for giving me the chance to deliver this speech. I am working as an assistant doctor at the Faculty of Cerrahpaşa Faculty of Medicine, and I'm also a PhD student at the same time. I will be talking about the microorganisms and their relation with schizophrenia and neuroinflammation. This is the outline of my presentation. We will talk about the early infection hypothesis and the epidemiologic trials that prove the infectious effect, neuroinflammation concept, and finally the relation between schizophrenia and neuroinflammation. There is the hypothesis whether the schizophrenia is actually a result of infection, and this hypothesis dates back to 1918, and in a book it was mentioned by Esquirol, and their causes can be infectious. That was the argument in the book. And then came the Kripalin, and he talked about the cause of dementia, and the infections in the body are made because of this, but the pathogens of them are causing the disease. They don't need to invade into the brain. And we have talked about the influenza pandemic in 1918, and then came the epidemics of psychosis, and the attention was paid on this area later on, and neurotrophic viruses were studied, and we began to understand their roles in the pathogenesis, and not only the viral infections but also the bacterial agents, and finally Toxoplasma gondii and parasitic infections have a close relation. When we look at the recent epidemiological studies, we see a Swedish study, and there were lots of people involved, participants involved in the study, 1.2 million, and there was a similar study from Denmark in 2014, and it was said that whatever the cause of the infection is, the admitting to hospital for infection can be a reason for schizophrenia, and it was found that there are some acute exacerbations in those patients. So, why should we shed a light on this etiopathogenesis and its relation with infection? We know that it's a chronic progressive disease, and if we shed a light on this area, maybe we can make use of some preventive treatments for schizophrenia, and shedding light on the pathogenesis will give us the chance to make an early diagnosis and early intervention. There is a multicentric study from 2015, and the biomarkers obtained from the peripheral blood were used, and you can catch the schizophrenia before its onset based on this approach. And the antidopaminergic drugs can be used for schizophrenia, but they are not sufficient. So if we can shed a light on the pathogenesis, then we can create treatments for these symptoms. When we look at the etiology, of course, we have the biopsychosocial model in psychiatry, but here we have the biological model that is important for us, and we should first focus on the genetics here. But in schizophrenia, genetic predisposition is not in line with the Mendelian inheritance, and a couple of genes and the environmental factors come into effect, and as a result we see schizophrenia. So it's not only related with the genetics or the environmental factors. Both the genetics and the environmental factors come together and produce schizophrenia. When we look at the environmental etiology, there are pregnancy complications, malnutrition, and city life. These are important factors. And when we look at the environmental agents, we are exposed to millions of them. I mean, we have microorganisms and we have infections that we have throughout our lives. So they all have an effect in this pathogenesis. Neuronal inflammation, of course, infections can be neurotrophic or not. They can go invade into the brain or not, but they show their effects on the brain through neuroinflammation. Therefore, the brain and the spinal cord are affected by the neuronal inflammation. And cytokines, chemokines, ROTs, and secondary messengers are the regulators, mediators, and glial cells, and central nervous system, endothelial cells, and peripheral immune cells are the actors that play a significant role. And we know that microglia is actually the cell that is orchestrating the brain and immune system. And on the left hand side, you can see the fluorescent staining of a nucleus, and also you can see its extensions and processes. And during the resting phase, the microglia, with all these processes, they actually audit the environmental tissue. If there is anything in the environmental tissue, there could be a neuronal damage or immunological activation, then this microglia is activated. And as you can see here up there, the cell body is large, and you can see the extensions are slow. So it is a phagocytosis now, and it releases cytokines, and for schizophrenia, it causes synaptic pruning. Once the microglia is activated, it has two pathways. The first one is M1. This is the pro-inflammatory pathway. The M2 is there as well. After the M1 pathway, the M2 pathway should be activated. If there is dominance of M1 pathway, then it will lead to a loss of neurons and synaptic damage. And also we have the priming, and you know that perinatal infections or maternal infections with maternal activation may affect the infection in the brain of the baby. So priming means that if you give repetitive triggers to a cell, then the cell will give more response. And in the microglia cells, there are three scenarios. It will just be deactivated, it will be hyperactivated because of lots of stimulants, or it will lead to priming. It will be silent. But if it is primed, and when it encounters the infection again, it will be hyperactivated. When we look at the relationship between schizophrenia and neuroinflammation, we know that metal infection, maternal immune brain damage, and also childhood infections. These are all important factors for schizophrenia, and they increase the risk for schizophrenia. And we know that the activation in microglia also increases the risk for schizophrenia in childhood. I will talk about the maternal infections, because if the there is maternal immune response increase. That means pro-inflammatory response is increased, and the cytokines here go through the placenta and activate the microglia, and they cause priming, and this causes microglial dysfunction, and it may lead to neurodevelopmental problems. And as a result of the synaptogenesis disruption, we can see impaired dopaminergic response, and in rats, we can see hippocampal size changes as well. How can we explain this? In some groups, the schizophrenia appears in the fetus, but sometimes it does not. So it is a dual hit maybe, because if you have any genetic predisposition for schizophrenia, for instance, then you may have schizophrenia if you are exposed to at least two triggers. And when we look at the other model, the response we give to stress is actually determined by our genetic code. When we look at the schizophrenia genetics, we have lots of locus divas trials, and the sixth chromosome MHC gene locus is very correlated with this issue, and it codes the complement 4A, and it is effective in synaptogenesis. And in the variants of this, we see that there is reduction in neuronal connectivity. And we have schizophrenia, the chromosome 22. This codes the claudin 5 protein, and in tight junctions we see this, and if there is any damage in tight junctions, then the blood-brain barrier has increased permeability, and with this increased permeability, it will be more sensitive to peripheral immune response. And this increased permeability helps us because we can understand M1 predominance in the brain. It's not only about the immune genes, but also we have non-immune genes that are related with schizophrenia. For instance, this one gene, and it is actually working in the neuronal skeleton, the neuronal body, and in the gene damaged rats, there is an increase in interleukin 6 levels, and the other one is a nuclear receptor regulator 1 gene, and this one is in the dopaminergic neurons in striatum, and if there is a damage, then there is an increased interleukin response. Neurotransmitters not only affect the genetic, are not only affected by genetic mechanisms, but also if there is a decrease in the cortical volume, then it may lead to subcortical dopaminergic disinhibition, and this leads to increased acute stress response. This one, mutant rats were exposed to perinatal infection, and this leads to increased glutamate response and increased dopaminergic response. This is the algorithm what I have tried to tell you. While trying to treat schizophrenia, we're trying to treat starting from the end of this pathway, but we should go back to the start. We have some agents, but they cannot pass through the blood-brain barrier, and they cannot affect the hippocampus or some other areas. And maybe we can have some agents so that we can have a better treatment for schizophrenia. Thank you very much, Ersed. And it is a very complicated topic, and thank you very much for all the information you have provided. Now we'll proceed with the last speaker from Istanbul Aydın University Faculty of Medicine, Dr. Özer Akgül. She is working as a microbiologist and she will be talking about the interaction between microbes and schizophrenia.
Thank you. Hello, everyone. First, I would like to thank to Ömer for their valuable invitation. I will be presenting the final part of this concept, actually. In the pathway to schizophrenia, we will check the impacts of microbes. And there are certain methods for prevention. I will be talking about that as well. Within the context of this presentation, I will be talking about microbiology and psychiatry, pathogens and schizophrenia, superorganisms, microbiome, the research techniques, microbiota, gut and brain access, which is a new concept, and we will check the reflection on new practices. As our colleagues have stated, microbiology and psychiatry relation dates back to 50s. However, toxoplasma is the first structure that we have observed. That toxoplasma resides in mice and it transmits to cats actually. It is a behavior changing parasite. The researchers show that if mice is infected with toxo and they are affiliated to the urine of cats. This is the cause of parasite, and the toxoplasma tries to complete its life cycle, and it is also related to prevalence, but in experiments they have just proved the relation. And when we check the study to microbiology and psychiatry, the number is over 4,000. It has some decades only. It is a very up-to-date hot topic. And it has been stated that there is a new term, psychobiome. So all the organisms which have certain impact on psychiatric disorders are called as a psychobiome. And this is now accepted as a new subfield for study. Why? Because in psychiatry and other medical areas, need to have evidence-based studies, and surely through the labs we can examine all these microbiomes and identify their impacts. I will be talking about pathogens. You do see there are lots of microorganisms. They are neurotrophic. In either way, they are either latent to the nerve, or they need to reach to the nervous system. Increased schizophrenia after infectious diseases is one sign for us. There are some parasites as well, but unfortunately there are not that many evidences proving their direct relationship. For bacteria, strep is one of them, and also fungus has certain types which has impact on psychiatric disorders. Stomatitis, as you do know, it causes certain infections, especially after organ transplantation, for example, and it has the feature to be in the latent of brain structure. These latent structures just leads us a certain information for the psychiatric disorders. There are 46 studies examining the relation. However, after the pandemic, there was this idea to prove the relation. CMV were studied a lot back in the 50s. However, now it is a research field once again to identify the pathway. CMV positivity cannot be claimed very strongly for its increasing impact on schizophrenia. However, there are some studies elaborating the relation. And in the left bottom side, you can see our studies. CMA positivity is related to schizophrenia, and it was statistically significant. For T. Gandhi, it is still suspicious in terms of psychiatric disorder impact. A CMV. And this is another parasite, Toxoplasma gondii. The discovery of that dates back to 80s, but in 50s. However, in 1953, science people had the idea that it could be related to schizophrenia. Toxoplasma has been examined experimentally, and in 1970, they claimed that it has impact on behavioral changes. This dates back to the mice studies. And in 2003, there is the Torrey Yoten et al. Group. They have published one study in 2003 and etiopathogenesis of schizophrenia also includes toxoplasma's impact, and it increases the risk for the disease. It is a very complex mechanism, but we can say that this parasite has a protozoid form cause latent infection, and that latent infection cause disorders in dopaminergic and glutaminergic abnormalities. So there could be a bipolar disorder as a result of these abnormalities. But to what extent there are differentiations? We do not know exactly. It has not been enlightened. As I've stated, there are lots of studies in this toxoplasma field. And year by year, it is the numbers increasing. But I will show you 3 meta-analyses and our latest study, which is the most up-to-date study in the field. First one is coming from 2007. 3,873 schizophrenia patients versus 7,000 control group included to this patient, and they had this meta-analysis calculation. And as a result, they state that toxoplasma positivity increases the schizophrenia risk by 2.6 folds. And another study dates back to 2012. 667 schizophrenia patients, 1,700 control patients, and it seems that the risk increased 2.7 folds. And this is the most updated current meta-analysis. 12,000 schizophrenia patients included 70,000 control group examined, and as a result, they do see that toxopositivity increases the schizophrenia risk by 1.8-fold. You may see the correlation as well. Both for the table may change in accordance with the country and the number of infection. However, there is a consensus over this idea. And this is our study. 170 schizophrenia patients and 120 healthy control group individuals included. We had a PCR test, which is quite sensitive, and toxopositivity increases the schizophrenia risk by 3.07. Fold. So we have identified that there is a very clear significant risk. So far, I've tried to explain the relation in between parasites and schizophrenia, but now we come to the microbiota. As microbiota, in the past we were calling it as gut flora. However, we do know there are millions of microbes, and there are 1.3-fold times higher microbes in our body when we compare it with our cell numbers. And we do live with 150 times a fold microbe gene then again, when we compare it with our own genes, so we may say that they are the host, we are the guests in these bodies. Microbiome could be explained as a metagenome of their functions and the environment. And microbiome is the most up-to-date definition. The functions are examined, what is the relation between the environment. They have this department now, and the impact level is quite high when it comes to this field's studies. It is not just the gene structure, but also the functions. The evidence levels are quite high, but it is not quite easy to perform such studies. You need to have a very sound hypothesis. You cannot just check it later on. You may not be able to add an additional group. It is quite expensive. I will tell you about the stages later on. And with this US dollar rates in our country, it will be quite difficult for us. ELISA and PCR tests could be performed individually. However, microbiome studies cannot be performed individually. It is effort intense study. And you need to be able to interpret the data after all the examinations. But surely they are doable with a good team. Microbiome analysis. It dates back to 1977. Psyche discovers that, and Sanger sequencing methods have been discovered by him, and we had the Human Genome Project. When it was concluded in 2012, we do see that there are 46 chromosomes, and there are such other chromosomes with the functions of that and this. Those were basic information. However, scientific improvements, scientific technology have improved through the years, and they've started microbiome studies in regard to the functions of that. And in 2019, they have reported that they are in the third stage, but haven't shared any results at this point. At a microorganism level, they are sequencing all of these and examining. There are 4 stages. I will be sharing that. In the first stage, it is a DNA extraction. This is the basis of DNA studies. In the DNA extraction, you should just extract the suspected sequencing, and you should identify the material, because you will be sequencing that material later on, the suspected material. And then you have this pure DNA. You will compare the suspected one with the pure DNA. And suspected microbiome environment. Within that environment, you will identify the standard example to identify which ones should be selected and which ones should be left over. You should add barcodes to the genetic tips of it. And these are the things that I know. These are the ones that I do not have any information in regard to. And sequencing is quite important. When we say it is a Sanger system, it was the first one. We had the second generation. In United States, there is this third generation sequencing opportunities as well. But in Turkey, we are in second generation sequencing opportunities. All these just perform work. Genome is quite complex as a molecule, and it provides you the opportunity to sequence all of it in just one attempt. The third generation has a much better sequencing option, but it is still the same mechanism actually. Bioinformatics provides us the examination of all this information. After sequencing, you just do not compare the suspected parts and the pure gene, but you should eliminate the irrelevant parts. You should have a relevancy as well. And in bioinformatics, we have analysis. On the right side, you may see the graphic. There are autolus. This is the microbiota analysis. For example, in schizophrenia, some increase, some microbiota increase, and some features decrease, and this color tables provides us information. And at the end of all these technological improvements, there is this new definition, new concept, microbiota gut-brain axis. At first, it was gut-brain axis, but now they thought that it will not be sufficient enough to explain the concept, so microbiota is added as well. In the first discovery phase, they thought that in the gut the flora has changed, and the flora change caused changes in the brain. However, it was not a one-way ticket. There is a mutual interaction actually in between gut and brain. The variety in the gut affects brain, and the brain changes affects gut. This increases, decreases, additions of new types, etc., are also associated with different diseases, epilepsy, neurodegenerative diseases, and psychiatric diseases are all included to the context for examination. And actually, they didn't just develop this relation, they also continue with the treatment. Microbiota transplantation is the technique used for that. There could be a pathology because of the decreased firmicinogen, and as an addition to the pharmaceutical treatment, they would like to replace firmicates, and they are hoping to adjust the situation. And there was a confusion. Do we have schizophrenia because there was a change in microbiota, or there was a change in we get a schizophrenia, and upon that we have a change in our microbiota? Neurotransmitters change upon the changes in microbiota, in flora, so there is again a dual mutual interaction. On the right side, there is a publication. It is the most up-to-date publication, and it says in a summary, there are lots of data by the way. Enterobacteria group, bacteria, decrease in schizophrenia patients, and there is an unclassified group in that enterobacteria group, and that unclassified group increases with the diagnosis of schizophrenia. For DM, for cirrhosis, for example, there are lots of researches. However, in regard to schizophrenia, unfortunately, we are not in a position meta-analysis. This is a very current hot topic for psychiatry. And this microbiota analysis lasts for 4 weeks in average. Just the lab site takes 4 weeks. This effort-intense work provides us translational reflections. You can just identify the situation clearly. If you are talking about translational reflections, there are 3 aims. The first one is biomarker identification. For schizophrenia, it is difficult sometimes, and if you have one biomarker to support your clinical view, then you may have the diagnosis, and it is quite important to have this. Even this effort to find the biomarker is a promising issue. The second purpose is therapeutic optimization, because you provide a treatment but you can't see any impact on the patient. But with this translational reflection mechanism, you may have therapeutic optimization, and you can also just discover a new agent upon the examinations. For cirrhosis, now we have completed all three stages. The three panels have been worked in full, and a drug group have been developed. There is one genetic element. It could be parasite, it could be fungus, or genetic variations coming from the parents. However, if we can work on the other issues, for example, if we can divert the situation, if we can reverse the mechanism, then we can prevent the development of disease. This has been published in Lancet Psychiatry, and blood test to diagnose schizophrenia, self-imposed limits, and psychiatry, the title of it. The author states that if there could be an identification of a biomarker for schizophrenia, we may just validate the clinical sensitivity, and together with biological markers, it would be possible to diagnose schizophrenia in near future. They have this idea, so most probably in near future, or in the future, it could be possible to have biomarkers on the diagnosis of bipolar schizophrenia, and in some other psychiatric diseases, to increase the sensitivity of diagnosis. Thank you so much for your patience. Thank you. Thank you, Özer.
Gerçekten
We really need a marker. We need actually many markers because we don't have a specific one right now, but in the near future, I believe we will have such markers coming up. This is the end of the panel. If you have any questions or comments, we can take them now. My name is Dr. Mehmet. Thank you very much. This was a very good panel. What I understand is that there are some microorganisms and they have some systemic inflammation. They cause some systemic inflammation, and this leads to a disease, and also results in a synaptic damage and sometimes causes psychotic problems. But Özer actually talked about the microbiota issue and fecal microbiota transfer. This is done for autism, as far as I know. Do you have any studies that show the efficacy of this approach? And clonazepam and olanzapine are the psychotic drugs, and you know that they are used for some eating disorders, and this eating disorder may cause some problems in the intestines as well. Do you have anything to say about this, about their efficacy and how to improve its effectiveness? Or Does it contradict with your thesis because it has some effects on the intestines? Thank you so much for the question. The first one is about fecal microbiota transfer. Especially for autism, they have very good results, and especially in autism. And I believe that pediatric psychiatry should be working on this more, and especially for the healthy individuals that they have more of it. And in autism spectrum disorder, there are some trials about this, and I don't know the details of this, but I have heard of them. And the other one is about the eating disorder, and unfortunately, this is done by a group of people, and you see that there are more bacteria or less bacteria, and you are given a diet plan, and it's kind of a commercial approach, but I can tell you that with some diets you can actually give some bacteria into the intestines, but you can go to the pharmacy and get enterogermina and buy it, and if we give it to intestines, it will be much better because bacteria can talk to each other, and if you give more of what you already have, then you will not be replacing the missing one. So you need to find the strain that is missing, and then you will be compensating what is missing there. On the other hand, I can tell you that it is a commercial approach. Thank you very much. Especially for children whose parents have such a risk, the children also have a high risk. So if you get such a measure, maybe it will reduce the risk of schizophrenia. Yes, maybe you can reduce the risk of schizophrenia, but I don't know there is any trial about this. But from a scientific point of view, it is logical. You are not dealing with the genetics, but you can manipulate the microbiota, and you can prevent at least one of the risk factors by doing so. Thank you. Any other question or comment? Hello, I'm from Erciyes University. My name is Gökhan Ünal. I would like to thank all the panelists for their presentations. I have a couple of questions. The first one is that, yes, my colleagues have already mentioned it. There are some reasons that lay the ground for schizophrenia, and prenatal infections is one of them, and they have a significant impact on the developmental stage of the children. And there are some approaches that try to reduce the inflammation, such as aspirin or some other agents. But what I understand is that these are having a mild or moderate effect, but none of the trials have shown a major effect on schizophrenia. And we know that it causes some developmental problems, and we know that when these children grow into adults, it's not possible to bring back the neurons that have been lost. Therefore, my question is that what should our approach be for inflammation? Should we have a prophylactic approach, or should we turn this into a real treatment modality? The second question is that about the microbiome analysis. Yes, there are lots of bacteria in in the intestines, and sometimes there are more of them, sometimes there are less of them. But what is the ideal level of microbiomes that should be in the intestines? Because it's not like a substance that is missing in your body. It's a huge cluster of bacteria. Therefore, what should be the target level? What is the ideal level? And how much can we achieve the ideal level? These are my questions. Thank you. Thank you very much for your question. Maybe regarding your first question, I can give an answer. At the beginning of my presentation, I gave an example. I told that this is just one or two pieces of a 1,000-piece jigsaw, and there are lots of other pieces, and it's not easy to talk about all the other remaining pieces because we cannot develop a prophylactic therapy for it. This is just a theory, but we have increasing proof about it, and we don't know what we will do about it yet, although there are many evidence coming up. So with regards to preventive medicine, I cannot tell you much about this regarding the current literature. Maybe I will say a couple of words about this, but I believe Özer will answer your question about microbiomes. Thank you very much for your comment. Should we give it during an acute attack or acute episode? Of course, there are some studies that show us their efficacy, but the drugs we give do not pass through the blood-brain barrier, and the receptor areas are mostly in prefrontal cortex, in hippocampus, and they are not binding to those areas sufficiently. Clozapine, for instance, we don't know its exact effect, but in those areas, it creates an anti-inflammatory response in the later stages of the treatment, and this is why it's effective. So we don't have an effective treatment for this area. However, these anti-inflammatory drugs give some good effects. Of course, it's not possible to talk about a real causality here because this inflammatory theory may be supporting the genetic predisposition, or maybe a patient with genetic predisposition can be a trigger for this process, or maybe it's strengthened by this process, so we don't know it clearly. Therefore, as mentioned by Ersel, there are lots of studies focusing on strengthening the microbiota, but maybe in the future there will be more promising results. And I can give the floor to Özer for the other question. Thank you very much for this question. What is the ideal form of microbiota? Yes, we have 7 bacteria groups, and we want an ideal group of them. And we have results from some trials. And the abundance between these 7 groups should be in balance. So if they are in balance, then the microbiota is at ideal level. Sometimes there could be fabricate lowering, or something else is increasing. So this causes dysbiosis, and we know the ideal level, I mean almost ideal level, but sometimes there is a shift in the axis, so this could be the reason because of the pathology. Thank you very much for this answer. Any other question or comment? If any, we can take them. I'm working on a publication on the cognitive dysfunctions after COVID-19. I checked it on 64 patients. I had a global assessment of them, and then we had some tests done and I checked their functions. There is an interesting finding in there, especially those patients who do not receive any treatment have more cognitive impairment. And then we looked at the ICU patients, and those patients received ICU treatments. This is the other group, and the other group is the admitted patients, and they received treatment as well. Therefore, this anti-inflammatory treatment may cause some neuron damage based on these findings. Thank you very much for this valuable support and finding. Thank you. Any other question or comment? I guess not. Thank you very much for your participation, distinguished colleagues. We have done our best to conduct this panel. Thank you very much for your participation. Have a nice day.